My name is Dylan, and I'm a 5th year at Clark. I graduated this past May with a Bachelor of Arts in Environmental Conservation Biology, and I'm currently a biology master's student. I work in Deb Robertson's Marine Biology Lab, where we investigate a group of enzymes responsible for assimilating environmental nitrogen into the cells of diatoms. Diatoms are a group of unicellular phytoplankton, and they typically live in marine environments. My project is specifically investigating whether the downstream region of two different nitrogen assimilating genes has a role in gene regulation through changing the stability of protein precursors, or mRNA. We have reason to believe that this region, called the 3' UTR, is crucial in allowing diatoms to rapidly downregulate specific genes depending on the availability of nitrogen. If more reduced forms of nitrogen are available in the environment, such as ammonium, the genes responsible for assimilating more complex forms of nitrogen are in turn downregulated. Diatoms are one of the greatest primary producers on the planet, and as a result, they are extremely important players in the cycling of carbon. Understanding how diatoms utilize nitrogen is an important step to further unravel their role in their marine ecosystems. Such research may prove to be increasingly important as ocean acidification dramatically changes our ocean water chemistry. In order to develop the materials necessary for my project, my work has heavily involved creating task-oriented plasmids through the cloning of small DNA fragments by PCR and bacterial genetic transformation. These plasmids, which vary in DNA sequence, will allow us to decipher how discrete parts of a gene may play a role in the regulation of their expression. Completed plasmids were transformed into cultures of diatoms through particle bombardment, and transformed diatoms were then selected for possession of the experimental plasmid DNA. Other lab work has included sequencing DNA and designing or modifying existing laboratory protocols or methods to fit the needs of my specific project. I like to get most of my lab work done during normal weekday hours, but I have been known to go into lab at night or on the weekends. Not surprisingly, it can be a lot of fun getting lab work done in the biology building when it's completely empty. You know, maybe play some music of your own choosing at a slightly higher volume as you normally would and have fun with it. As a master's student, I work on my own time and work on my project when it works best for me. Such a flexible schedule is a luxury that I really appreciate. Because my professor likes to take a hands-on approach in the lab, I receive a lot of direct instruction one-on-one with her. This is the best way to learn the techniques of molecular science, and as a result, my abilities as a student researcher have grown greatly. I also work a little bit with the postdoc in our lab, as well as my fellow graduate students. We like to help each other out whenever possible. I also might work a little bit with some undergraduate students so they can continue my research project after I leave Clark. Working in a lab has been the highlight of my undergraduate career at Clark, and I'm really glad I pursued this path. Directed research has allowed me to better understand the techniques that scientists use to conduct their own research, and has made me a better science student in the process. If you're serious about studying science, be it biology, chemistry, or physics, you should really consider joining a lab at some point during your undergraduate career. My directed research at Clark has set me up with a fantastic array of lab techniques and research methods that will allow me to pursue a PhD in biology. While I feel confident that I could find a related job in a lab or at a research institute, that's not really for me right now. Rather, studying at Clark has set me up well to become a conservation biologist, which is what I want to do.